Well, thank you all for giving me a little bit of time to speak to you guys today. It's always a pleasure to come down here. And what I'm going to talk about today is work I've been doing for the last 7 or 8 years in cell line models of chemical and chemotherapy exposure. So I'm going to give a bit of an overview of what we've done to cancer with cancer cytotoxics, and then also mention an application in environmental chemicals, about 180 of them in partnership with the NIEHS. So, in general, right, understanding the variability in an individual response to drugs or chemical exposure is a key goal of pharmacogenetics and toxicogenomics. And goals of genetic mapping, right, where GWAS chips are now incredibly cheap and easy to do, and there are several goals that we hope to attain in doing GWAS mapping of drug or chemical exposure. We would love it if we can find efficient predictors of response, if it's pharmacogenomics and efficacy, toxicity, whatever is clinically relevant that might make a good biomarker. But we're also interested in dissecting and understanding the underlying genetic etiology of differential drug and chemical response. And we're happy to get interesting results under either one of those. And I actually am going to walk you through 2 examples, one of where we've found what we think is a potentially successful biomarker, and one where we're nowhere near the biomarker stage, but have uncovered some interesting and novel biology. So a lot of my work is in pharmacogenomics, and there's several things I complain about and whine about in doing pharmacogenomics instead of common complex human genetic trait mapping. We often have major study design limitations, so most of my pharmacogenetics work is limited to dealing with clinical trials. There are a number of limitations in that. Clinical trials never include family-based data. You're often really limited in sample size, right? If I'm working in a phase 2 clinical trial, these might be the only 120 people in the world that have ever gotten this drug. And limitations in replication, right? Again, if I'm doing work in a phase 2 clinical trial and that's everyone that's ever gotten the drug, how do I replicate my findings there easily? This also limits, and sort of one major limitation, I think, in both pharmacogenomics and toxicogenomics, is we have to make a leap to a couple of major genetic assumptions. So I trained in genetics, right? I would like to think everything has a genetic etiology at some level, but if I'm going to use association mapping, my trait needs to not only be genetic, it also needs to be heritable. And sort of without access to family data, I can't robustly test that assumption. So I don't know that sort of investment in GWAS mapping is going to be worth it. So because of these limitations, my group has been using, right, sort of if I'm a geneticist, I assume everything is genetic. So in response to these limitations, my group has been working with immortalized lymphoblastoid cell lines as a model for differential drug or chemical response for a number of years now. Like I said, we've used it to look at cytotoxic mapping for a number of chemotherapies. We started with the cytotoxics. We're basically trying to eventually do every FDA-approved chemotherapy as the assay technology will allow. And as of today, we're up to 45 drugs that we've been able to assay. We started with our cytotoxics, then moved on to monoclonal antibodies. We're currently working on tyrosine kinase inhibitors. We're basically reproducing drug development, sort of the oldest drugs that have been off patent and are cheap up to these newer sorts of therapies. Also, as I said, I like to say I'm a geneticist. I'm also a statistician, and there are lots of fun methods problems in sort of evaluating these things at really high throughput levels. And as I mentioned, we've also expanded this model for chemical exposure as well. So when I say we've got a model, what sort of assays do we actually use? For the most part, the chemical exposure is an exception to this. But for the most part, we use Alamar Blue assays. So Alamar blue is a dye that gets uptaken by cells that are actually alive and metabolizing, and doesn't get uptaken by cells that are dead. So we use 6-point dose response curves in immortalized lymphoblastoid cell lines. And here's an example of the type of response you expect, right? These are all cytotoxics. So you expect as the dose goes up, the amount of viability decreases. So the more cytotoxic you put on there, more cells will die. And that's what you expect. Like I said, we're currently using this to model 45 chemotherapy drugs. I'm going to talk today about what's come out of really the 31 drugs that we did first, because we've had time to functionally follow those up, and I want to show you some of those positive results. So we typically start with Ceph cell lines that have an extended pedigree structure. So we can quickly run these assays in family-based data to check heritability, see if it's reasonable for us to follow up. Then we follow up with mapping in unrelated cohorts. Our first experiments were done with a cohort from the Children's Hospital of Oakland Research Institute, a gift from Ron Krause. That is a Caucasian cohort. We are now doing all our experiments in the 1,000 genomes panel. We've got a much more diverse population. And one of the efficiencies of this system is we only ever use cell lines where genotyping data has already been produced or publicly available. So for the cost of just doing the dose response assays, we can do GWAS mapping in these cohorts. The environmental chemical experiment I'm going to talk about was done in the 1,000 genomes panel as well. Like I said, and there are lots of methods challenges along the way. I want to show you a couple of sort of typical dose response curves, because I want you to understand the magnitude of what I mean by when I say we observe differential response across the population. So this is actually two different Ceph cell lines. So these are actually related individuals. So I'm not even getting towards sort of differences across subpopulations, etc. And what I'm showing you here is two different Ceph cell lines and their response to docetaxel, one of the drugs. We repeat these experiments in a number of different ways. What I'm showing you here are both biological and technical replicates, where we do 4 replicates within a plate, and then we repeat that 3 times. So you can also see how really reliable these assays actually are. So here's an individual that's highly sensitive to docetaxel, and here's someone that is much more resistant. So this is the sort of differential response to these drugs and chemicals that we actually observe. So we started with 29 classic cytotoxics, and I just wanted to show you the magnitude of heritability that we're seeing with these drugs. And the chemical exposures looks very, very similar. And what you see, we have control samples on our plates, and I realize this is probably too small to see, but we have heritabilities typically on the order of sort of 12% up to over 60%. So we're in the range of sort of the heritability of common complex diseases. And I will say there's a strong correlation between those drugs with the highest heritability and the success we're having in actually finding signals, which is reassuring. So like I said, then we follow up with a GWAS association in unrelated individuals. I mentioned the CHORI population before. It's 520 samples. Now that we've switched to the 1,000 Genomes Project, we've been able to increase those numbers a little bit. And this first study was done with a 650K Illumina chip. So as I mentioned, there are lots of fun methods challenges hiding in here. And one of the sort of things we've tried to tackle is how do we accurately fit dose response? Curve fitting is a a nasty statistical problem to do that sort of nonlinear dose response. And with such high throughput experiments, we wanted to minimize the number of assumptions that we were making. So other people use the cell line model as well, and most people will fit one aspect of the curve. So if you're doing a 4-point dose response model, you've got things like the EC-IC50, or you could pick another one, right? The dose it's required to get to 50% kill. A lot of other groups have used the hill slope, so what actually is the slope of that line as sort of you get that drug response. And these choices make lots of assumptions. And as a geneticist, I certainly know, sort of in a high throughput way, any model you can dream up, plus several that you can't dream up, are probably the truth of how sort of genetic associations actually work. And I've drawn a quick little toy example here of different types of genetic associations. So if differential drug response is associated with SNP level variation, right? We have 3 categories of SNPs that I'm showing here in black, red, and blue. And we could have true associations that actually affect the min of the dose response curve. What is your sort of the floor effect in these assays, and is that what changes? We also could have situations where the sort of the genotypes cause differential IC50s, and or you could have sort of models where that hill slope is actually changed. And when we're starting to do dozens of drugs across thousands of individuals, we have cases where we see each one of those. So we wanted to come up with a way to do more robust modeling than actually doing sort of fitting one of those parameters and then doing association. And so we've chosen a Mancova approach, basically, so it's not methods development as much as correctly applying traditional statistics to a large setting. And this approach we find minimizes model assumptions, and we've supported and maintained software packages to do sort of Mancova modeling and genome-wide association studies for a number of years. So now I'm going to jump to the results from the first 29 cytotoxic drugs that we looked at. So here's sort of a boring, dry list of all of the drugs that had suggestive, at least suggestive associations with them. So we've got the drugs listed here. Come back. The drugs listed here, then what chromosome, and then the RSIDs. And we've got the negative log 10 p-values here. What you'll notice is most things are at sort of middling suggestive level. There is one that really, really stands out, right? So we got an association for temozolomide that was well into genome-wide significant territory, so it was the obvious candidate to actually go and follow up. So here's the Manhattan plot for temozolomide. That is a dream Manhattan plot, right? That is everything you want. One clear, beautiful signal. It got even prettier when we started looking into the underlying biology. So this is going to be my example where we haven't discovered new biology, but do think we've found a potential biomarker here. So this beautiful peak is in a gene called methylguanine methyltransferase, or MGMT. And actually, MGMT has been known to be associated with temozolomide efficacy. So temozolomide is used in brain cancers. And sort of it had been shown back in 2005 that differential expression of MGMT in brain tissue is a good marker, is strongly associated with temozolomide efficacy. That's interesting biology. Brain tissue doesn't make a great biomarker. People don't really like to get that tested. So we were excited to think, well, if we've got this kind of signal at the SNP level, could we explore using the SNP as a biomarker? The biology kept sort of matching. We then took, within that peak, we have baseline expression for these LCLs that we're using. So it's not drug exposed expression, but baseline expression. And we were able to show that, and this matches the public literature as well, that lead SNP in MGMT is an EQTL, and sort of the AA genotype, basically in an additive fashion, the number of A alleles you have is associated with increase of MGMT expression. MGMT is a repair enzyme. It actually repairs methylated cells. So basically it will repair cells as the chemo is trying to kill it. So then we associated that sort of SNP with viability, and you see the pattern that you would expect, that as you have increased expression of a repair enzyme, you have increased viability because you don't get as much cell death. So we followed this up with wonderful collaborators at Moffitt Cancer Center. We started with a preliminary data set, and I'm happy to update from a call this morning the results of a much larger follow-up of this. So we started with a pilot study of about 400 patients with high-grade glioma. The standard of care for glioma is resection, radiation, and then temozolomide. We pulled 7 SNPs that were in sort of linkage disequilibrium within that peak, right? Not wanting to entirely bet that it was the lead SNP there. And what we found is this is Bonferroni corrected for those 7 SNPs, that sort of there is a strong association in males, not females, of sort of the SNP with long-term survival. And the association is in the direction you would expect. If you have the SNP that increases MGMT response, you have decreased survival. The drug doesn't work as well for you. Like I said, I had a phone call this morning where we finalized glioma is an incredibly rare tumor, thankfully, but it makes it quite hard to get power to follow these things up. Again, the collaboration at Moffitt, they also had a sample of 1,800 gliomas, which is just amazing to be able to do. So we followed this up, and we get the same signal in that 1,800 as we do in this pilot study with increased significance. So we're seeing the exact same trend of sort of your SNP at MGMT and overall survival. Again, we also see the same difference with males and females. This is our preliminary analysis. We're going to have to do a lot more to try to understand that gender effect. Our first hypothesis is it's actually compliance. Women are much better at taking their medication when they're told to take their medication than men are. So we're going to start with that as a candidate to pick this apart. But there might be actual differences in the underlying etiology in the two genders. So we'll spend a month or so trying to pick that apart further. Next, I'm going to talk about an example success in the other direction, where we actually found some interesting biology. So far, we've done 2 monoclonal antibodies. Here, we use the same Alamar Blue assay, but the response is a little bit different. Because these 2 monoclonals that we've looked at, rituximab and ofatumumab, target CD20. So there's actually not sort of dose response curve to monoclonals. We apply this to the saturating dose and basically look at the maximum response to the drug. And again, taking our own advice, we started with the Ceph pedigrees to see if this is heritable. It's also, the Ceph pedigrees are where we fine-tune dosing for these drugs before we launch anything large scale, etc. And we didn't yet have genome-wide association data on the monoclonals, but since these are the Ceph pedigrees, we can peek and do linkage mapping. And we couldn't help ourselves. We peeked and did linkage mapping, knowing that we're not particularly well-powered. But what we found was these are the linkage peaks that were consistent between rituximab and ofatumumab. So there were 2 peaks for follow-up, chromosome 3 and chromosome 12, and it's the chromosome 12 peak that was, like I said, almost overlaid for those 2 drugs, so that's what we chose to follow up. When you do linkage analysis, you get dozens or hundreds of genes instead of a localized signal. So we tried to narrow that down even further by actually looking at gene expression data. So again, we had baseline gene expression for these Ceph cell lines, and we associated baseline expression with that maximum sort of viability response. And again, here, after a Bonferroni correction, we had one gene that stood out. So we go, great, one thing to follow up here, and that gene was CBL-B. It encodes an E3 ubiquitin ligase. It's involved in T cell and B cell receptor downregulation. CBL-B loss promotes autoimmunity. And that's really all we can find. CBL-B does come up in pharmacogenomics in general, but had never come up with sort of monoclonal or any chemotherapy or cytotoxic response before. So we're like, maybe this is something a little bit new and interesting. So we followed that up with functional validation. So both rituximab and ofatumumab's target is CD20, as I mentioned. We first did knockdown experiments to show that if we knocked down CBL-B, did that change overall response? The answer was yes. Then we wanted to see what happens to the expression of CD20 if we knock down CBL-B. So we did that experiment and first looked just for overall expression of CD20 when you knock down CBL-B. We got nothing. I mean, the p-value is in the 0.9s. I don't remember what it was, but it was not even close to trending. So then we actually decided to follow up and not look at just sort of overall expression, but look at the localization of expression of CBL-B. And that we saw a dramatic result. So what I'm showing you here is we repeated this experiment in 4 different cell lines. 3 of them are LCLs. This Raji cell line is actually a B-cell lymphoma cell line, which is perfect because sort of rituximab and ofatumumab are given largely for your blood-borne tumors. So we tried to also get a cell line that was more relevant to sort of the correct tissue. What you see here is sort of the wild type here, so no knockdown of CBL-B. And here is staining results of the knockdown of CBL-B. And what you see is, again, the overall expression hasn't changed, but the localization has. We get these tight clumps, is that a technical term? We get clumps of CD20, and as we follow up, the CD20 actually doesn't reach the cell surface, so it remains in clumps and doesn't reach the cell surface. So we've got a good hypothesis of why that would change sort of your response to these CD20 targeted monoclonal antibodies. And that paper just came out this month. Where, like I said, this is the first time we've shown that CBL-B is related to these anti-CD20 drugs in any way, or that CBL-B is related to CD20 in any way. If you go to pathway sort of graphs, that's never come up. So this was an exciting sort of demonstration of GWAS being able to find truly novel biology. Now I want to skip back to the toxicology application where we're looking at environmental chemicals. So this was in partnership with folks at UNC, the National Toxicology Program, and NCATS at NIEHS. Again, we used 1,000 genomes panel, so that gives us a little bit over 1,000 cell lines that we can look at, and this gives sort of a diverse subset as well. We've got representation from around the globe here, which is exciting. We did this for 179 chemicals. And in this study, we were actually asking 2 questions. First, for these 179, we were asking the question, is this chemical cytotoxic? And there were some that were not cytotoxic to the LCL. And then if they were cytotoxic, can we map sort of genetic predictors of differential cytotoxicity? This is figure 1 from this paper showing you the scale of what all had been done before with these LCL models. So this Brown et al. Study was the 29 drugs with the 520 cell lines. So we've got number of chemicals or drugs shown on the x-axis, and then the y-axis demonstrates the number of individuals in that study. So a few people had looked at large number of chemicals, but in very, very small cell lines. And then sort of this study represents by far the largest study in both terms of number of chemicals and sample size being included. So for this, I'm going to go over these results very briefly, because you can imagine with 179 chemicals, a lot of things came out. So I'm going to give you sort of the most consistent and sort of interesting main results. So we analyzed this using that Mancova approach. Our software package is called Magwas, Mancova of GWAS. So we used that as our analysis method. We did heritability as well. We did also include the Ceph cell lines, and then we did, for the unrelated cell lines, we did marker-based heritability estimates, and we saw results that were very, very similar to the chemotherapy kinds of results. There were a large number of these chemicals that had heritabilities over 30%, and up to, I think, 55. We also looked at clustering by racial groups. We performed pathway analyses, etc. Demonstrated racial differences, in this case IC10s, sort of your point of departure instead of the mid-range point. And like I said, I'm just going to breeze through the results a little bit here. This paper is out. Fred Wright is the first author. Associations suggest important roles for variation in membrane and transmembrane genes was the most consistent theme we had. And this is the same types of genes that have been shown before for chemotherapy response. So these seem to be very just general cytotoxic response genes. They are not exclusive to chemotherapies. Several chemicals, sort of the gene that came up the most consistently was SLC7A11. Which has previously been associated with chemotherapeutic resistance, both in cell lines and in human samples, which, like I said, reinforces sort of the use of this cell line model. We did use public RNA-seq profiles, again, baseline expression, not chemical exposed expression, and it showed evidence of sort of association between basal transcription and cytotoxic response. And again, membrane localization comes up as the most consistent theme in this. Showing you a little bit some of the Manhattan plots, and you see we get strong signal, right? We've marked here at the suggestive level, but we get a number of signals that I would consider to be genome-wide significant. So what I hope I've convinced you of is that LCLs are a promising model for dose-response mapping. It allows for research that's not available and possible in human subjects. The high-throughput nature of these, right, as we expand, so 179 chemicals by almost 1,100 people, certainly counts as high throughput. It was done in NIEHS's facility, which actually has a great room. They've actually repurposed robots that used to work in the auto industry are doing these assays for us. So they're robots that are getting a second life. They're not building cars. They're now helping us do these. Typical association methods may not capture the full results. So we have, of course, compared sort of using a more traditional method where we just fit EC 50 or 10 or hill slope, and you do not see sort of the dramatic results that we see here. So I'm confident that our analysis approach is good. We've also done clustering on response by chemical or drug class, and we can recapitulate very consistently chemical or drug class, looking at differential expression or differential response in these. And we're learning, for the most part, this dose-response trait is as complex as any other complex trait. We've gotten a few sort of screaming examples, but it looks to be that sort of differential dose-response has a complex etiology, much like any other trait we try to look at in humans. What we are currently working on, so we've got data in and initial analysis on tyrosine kinase inhibitors. We're getting extremely consistent signal in a gene called NFAT5. In our tyrosine kinase inhibitors, we've done 18 TKIs, and this signal comes up at a 14 out of 18 of them, and we're doing the functional follow-up work now. We're trying to use the cell line models, I said, for the PD-1K inhibitors. We're getting some technical challenges in actually using those newer drugs, but I'm confident my wonderful collaborators will be able to work it out. And we're exploring other methods to interrogate this data, doing pathway-based approach, looking at cross-heritability for drugs and chemicals of very similar type, and then sort of also work a lot in finding gene-gene and gene-drug interactions in these data sets. Sort of methods that have come out of this, we've done a lot in sort of actually dose-response curve fitting and using evolutionary computing to actually do that with making minimal statistical assumptions. We've advanced sort of approaches in permutation testing as we've done this. So there have been lots of fun methods things that have come out. The last thing I'll mention here is the other direction that we are going is actually looking at mixtures in both the chemotherapy and the environmental chemical set. Right? Chemotherapies are rarely given alone. They're typically done in cocktails, and certainly environmental chemicals are rarely given as single sorts of entities. So we want to be able to start modeling and maybe even mapping that. So I'm going to talk briefly about some preliminary results that I'm really, really excited about in the combination work. There are also plenty of methods development challenges in doing synergy that I know I look overly excited about, but the nerd in me loves them. So things like the most commonly used method for synergy measurement is the Chow-Talele method that produces a combination index, and I'll show you, I could talk more about that if you're interested, and by far it's been the dominant method in the field for 25 years. There has been no way to do inference on that, so you can quantify this combination index, but I can't put a p-value on it. So you can say, yes, the null in Chow-Talele is a combination index of 1. I can say it deviates from 1, but not if it significantly deviates from 1. I've had a very, very talented graduate student that we've got a paper in submission now that actually adds inference to that. So we can say this is a significant deviation, not just an observed one. And we've launched these assays in a pilot experiment. So we looked at 8 different drugs in combination, and we picked, so these are our chemotherapy drugs. So we picked these for combinations that have been shown to reliably have synergistic effects in vivo. So for our pilot, we tried to pick combinations that we knew would have an impact. And I don't know if you can read that, but it's your usual suspects in chemotherapy: docetaxel, gemcitabine, carboplatin, some of the most commonly used chemotherapies in the field. We did 7 different combinations of these, so we have assay results for both the monotherapy and then the combination therapy. And again, we started with pedigree based. Can we actually quantify synergy in the LCLs? This will be the first study that's actually quantified it. There was one group that in late December put out some results on synergy in LCLs, but they didn't even fit the Chow-Talele. So can we actually look at the magnitude of synergy in these cell lines? And then we wanted to test the hypothesis of, is synergy or antagonism, right, its opposite, is that actually heritable? So can we see it in LCLs, and does it look like that's a genetic trait? And the answer was yes for both of those. So the heritability of synergy is a little bit lower. Now keep in mind here, when I'm estimating synergy, it is not, or estimating heritability, it is not the heritability of total drug response. It truly is the heritability of the non-additive effect. So this is purely of the synergistic effect. And we actually see heritabilities, our top one's a little over 25%, but we see things in the 15 to 25% range pretty interestingly. We also took a sneak peek at linkage results for these because I can't help myself. And one interesting finding we've seen, knowing that this is underpowered and maybe anecdotal, is we see differences in the overall etiology of the monotherapies versus the combination therapies. So if you look here, just a couple of examples. We have peaks that come up in the monotherapy. So this is paclitaxel alone. This is the combination of paclitaxel and epirubicin, and this is epirubicin linkage, and you see that there is sort of a peak here that comes up just for paclitaxel, but doesn't come up in the combination. And we have the opposite examples as well. So here we've got a peak that comes up in the combination therapy that doesn't show up in the monotherapies at all. So we're pretty excited about this. As I said, it's a pilot, but we're excited that this sort of may be a fruitful direction to go with the cell line models. And I will stop here in just a couple of moments. So hopefully I've demonstrated sort of that this is a useful model for chemotherapy and environmental chemical dose response mapping, and sort of have mentioned briefly that sort of I think methods development and biology should be hand in hand in sort of these approaches. I have to thank all my collaborators, and I'll stop there.
Hi, super cool work. So many questions because this is really cool. The first one is, you are starting out by doing heritability analysis with the Ceph Utah pedigrees and then doing linkage with those. And then you're going to an association-based approach in unrelateds. So you're going to miss any loci that have allelic heterogeneity. I'm wondering, have you gone back and done any conditional linkage analyses back in the pedigrees to see how much of that original heritability is explained by the SNP that you identify in the GWAS?
Yes, we have gone back and done that, and it's about the same magnitude of what you get in other complex traits. So that MGMT peak, for example, that is so gorgeous, it explains 5% of the heritability, and all of our other peaks explain between 1 and 2%.
Does it, so I do arsenic toxic kukinetic genetics, and with our main linkage signal on chromosome 10, the index SNP for the main known gene there explains about half that conditional linkage peak is like half as big. Did you do any locus-specific linkage conditional on the SNPs, or was it just kind of the overall heritability conditional on the SNP that you looked at?
We did both. So we did actually do conditional linkage looking at that. And just to let you know, arsenic isn't included in our panel of chemicals.
The arsenic trioxide, or?
The what?
Arsenic trioxide?
Yes. So I would love to follow up.
And then my second question is, I noticed that, and I think this is really interesting, the monotherapies were just suggestive linkage, but the synergy was actually significant. Can you speculate on why? Because that's so cool.
I know. I don't know why, right? Maybe that implies, and that was the case across all of our combinations. So we saw that sort of pattern. I presented paclitaxel and epirubicin, but we saw that pretty consistently across all of them. The other interesting thing that I didn't show here is we actually see a range of synergistic effects that go from synergy to pretty extreme antagonism, which is really, really fascinating. So it's possible that these sort of genetic variants actually might even predict whether that drug combination is synergistic for you or if it's antagonistic. But we're still in the pilot stage, and we're getting started.
One follow-up question. Mm-hmm. So you flagged that transporters seem really important, and that's kind of what we're seeing too. Are those, in the synergy ones, are those things that are popping out transporters that are shared or competitively inhibited by these things, or do you have any speculation? I know it's pilot.
We haven't looked. Like I said, this grant got renewed last year, and synergy is going to be a major focus. So over the summer, we'll start running the GWAS stuff on synergistic combinations in the 1,000 genome cell line. So I've sort of resisted looking at the linkage peaks for now until we get that data later in the summer.
Have you looked at statins at all, and the different statin types to see how they?
No. In the cell line models, we haven't looked at statins. I do look a lot. One of my other major projects has been pharmacogenomic mapping in the Accord clinical trial. I don't know if you're familiar with it, but large clinical trial of type 2 diabetics that is the most expensive failed clinical trial in NIH history, so why not do genetics? And we've done a lot with statin mapping in that clinical cohort, but we haven't done it at all in the cell lines.
All right. Thank you, Allison. Thanks.